All right, fellow scientists, welcome back. So today we're going to talk about something called gene therapy. So this is actually our last topic for biotechnology, and this is going to be our very last video for the flipped classroom. So to celebrate that, I'm offering a small extra credit opportunity on the quiz that we're going to take tomorrow. If you write the word pickles down at the bottom, big or small, or doesn't matter, it just has to be there, I will give you extra credit. Now, here's how this is going to work. I have 105 students on my roster, and so there's 105 points possible. Those points will be divvied up among the people that write pickles. Okay? So if all 105 students write pickles, then everyone gets 1 extra credit point. If only 50 students write pickles, then everyone gets 2 point something extra credit points. If only 25 people write pickles, then, well, you get the idea. So it is in your best interest not to tell other people who haven't watched the video, who haven't done the hard work that you're doing, to write pickles. So anyways, on to the topic of gene therapy. So gene therapy is, we're going to define it as curing a genetic disease. Curing a genetic disease. And before biotechnology, this was unheard of. I mean, your genetics are your genetics. They're passed down from your parents. They're what you're born with, and there's no changing them, right? So again, we're not going to talk about the tools, just like we didn't talk about the tools of cloning, because there are, lots of tools and very complicated. But the basic process of gene therapy is fairly straightforward and fairly simple. So we're going to use the example of someone who has sickle cell anemia. Now, sickle cell anemia is a condition where the hemoglobin in the red blood cells doesn't form right. And so normally, red blood cells, they look kind of like this. They're actually donut shaped, right? And they're the cells that carry oxygen from your lungs to your cells, to all the cells in your body, and then they carry CO2 from your cells back to your lungs so that you can exhale that. So normally they look like this. But if a person has sickle cell anemia and that hemoglobin is misshapen, you look underneath the microscope, and literally what should look like this looks like this, right? And so this causes all kind of problems. They can't get enough oxygen, and these tend to clump in the capillaries and the small little blood vessels in your cells or in your body. These tend to clump a lot easier than these. So we're going to say that this person right here, forgive me for drawing stick figures, but that's about the limit of my artistic ability. We're going to say that this person right here has sickle cell anemia. We'll abbreviate it SCA, sickle cell anemia. And of course, this person is not happy because they have sickle cell anemia. So they have pain and they can't get enough oxygen and everything that I just talked about. Then we have a person over here. Now remember, sickle cell anemia is caused by a defective gene that makes hemoglobin, right? And so it's a genetic disease. So you can't take antibiotics for it. You can't do anything except gene therapy. So then you have a person over here, and this person does not have sickle cell anemia, right? So what you do is you extract some DNA from this person, and then you're going to, because we have the human genome sequenced, we know where the gene that makes hemoglobin is, and we know where the restriction sites, so in areas where restriction enzymes can cut, we know where the restriction sites are. So, and again, this is more complicated in real life, but the basic idea is that you use a restriction enzyme to cut out the gene, and then you put this gene into a virus. Wait, Mr. Leo, I thought that viruses were bad. Well, not necessarily. Most viruses, as we know them, are bad, but a virus is just a delivery vehicle, right? It's like a Suburban. The Suburban can be carrying bad guys, and of course that's not good, and that's what viruses normally do. However, a Suburban can also be carrying good guys. So this is the gene for hemoglobin. Right? So the gene for hemoglobin, we're going to take that and we're going to put that gene into our virus. So by putting this gene in the virus, now we're making the Suburban carrying good guys instead of the Suburban carrying bad guys. And so then what we do is we infect this person who has sickle cell anemia, we put this virus where the gene would normally be. So normally people make hemoglobin in their bone marrow. That's the inner part of your skeletal system. So we would put that virus, we would make it attack the bone marrow cells, and so it's going to insert this good hemoglobin gene into the bone marrow of this person, and the idea is that then, because this person has a working copy of the gene that makes the protein for hemoglobin, then they will no longer have sickle cell anemia and they will be happy. Right, so it's kind of, this drawing is very simplistic, and like I said, it's a lot more in depth and a lot more rigorous. But this is the basic idea. Now, 2 articles for you. First article is from 2012, and this is talking about how gene therapy has been used to restore sight in people who were born with a genetic defect that causes them to go blind. So if you read this, you can Google this if you want to, or if you want to pause it and read it. Basically, these people suffer from a condition known as Leber's congenital amaurosis, and so they're born and they can see, but then as they grow older, then they slowly lose their vision until they're legally blind. The reason why they lose their vision is because they have a genetic mutation in one of the genes that makes an enzyme. Now, I won't get into exactly what the enzyme does, but suffice it to say that that enzyme is crucial. So what this study did is it took this gene, RPE65, and it put that into a virus, and they injected the virus into the eyes, well, actually into just one eye of 12 patients, and 6 of the 12 patients, where is it? Yep, there you go. 6 of the 12 patients improved for that eye, so much so that they were no longer legally blind, which is awesome. So then the same researcher did the other eye to those 6 who responded well, and 3 of them, in both of their eyes, they are no longer legally blind. So really, really cool stuff. Another version of gene therapy is to take out blood cells, and this is mainly being used to fight leukemia. And as you can see, this is 2013, so just last year, at the end of last year. And it's actually it's in what we call human trials, right? They know enough about this, and they think they have it down, that they're actually using this to potentially cure humans, which is, really, really cool. So basically what you do is you take out blood cells and you use a virus to put in a gene that programs those blood cells, those immune system cells, those white blood cells, to kill or to target specific cancer cells. So you're basically like giving the blood cell a mission. You're giving the blood cell attack instructions. And so then once those blood cells have those attack instructions, then they put the blood cells back into the patient, and hopefully, if all goes well, then the red blood cells will do what they're supposed to do and fight the cancerous cells, and then the patient will no longer have leukemia. And it's been remarkably effective. I believe up here, where is it? 120 patients given treatment. One study, 5 adults and 19 of 22 with cancer, acute lymphomic leukemia, were cleared of the cancer, were in remission. So really, really cool. So that's gene therapy, and that is the last video of our flipped classroom. Thank you very much for sticking it out to the end. Remember, write pickles and don't tell anyone.